Hey guys, thank you for listening to my thesis proposal. I've chosen topic 2, and my position is that I believe we can investigate the cognition of animals and safely infer about our own cognition to see how it evolved. Comparative psychology is highly popular, and it seems like scientists will pursue this project anyway. However, the more interesting philosophical point is to imagine what the impediments will be. That is, how are we to know and through what methods can we verify or be confident in that we perform such phylogenetic inferences appropriately? I'll examine this question in 2 parts. In part 1, I want to be sure we are clear about the target of explanation. What is cognition to comparative psychologists, and how could their anthropomorphized assumptions affect the evolutionary study of it? In part 2, I will sketch out a methodology from McLean and colleagues called the phylogenetic comparative approach, which hopes to effectively integrate comparative psychology and evolutionary biology to answer such questions. But firstly, some motivation. In The Descent of Man, Darwin claimed that the difference in mind between man and the higher animals is certainly one of degree and not of kind. However, a kind of dilemma is established. Firstly, it does appear more evolutionary parsimonious to assume continuity given greater relatedness in explaining a trait's evolution. A suite of comparative ethology research has defended this very claim by Darwin. Alternatively, though, we're struck every day with evidence of human cognitive uniqueness that suggests we are different and discontinuous with our closest relatives, the great apes. Our language, self-reflection, or theory of mind seem to be the product of a major cognitive revolution, both at the functional and representational level of cognition and brain. I believe this Darwinian tension of either proving continuity or discontinuity between humans and non-human animals sets the motivation for a large portion of comparative psychology. It's then prudent to ask, where could the philosophical and methodological worries arise from such commitments? A key limitation to explanations provided in comparative psychology is what Shuttleworth describes as the bias towards proving animals are clever, but only in human-like ways. There is considerable evidence in the literature discussing how certain non-human species demonstrate instances of communication, tool use, novel problem-solving, and planning behaviors. However, the explanations for such behaviors has been provided in human-like mentalistic terms and processes, instead of more simple cognitive processes like associative learning and species-typical predispositions, which would have far less phylogenetic implications. To provide this more simple approach or perspective is considered killjoy, even as such an explanation may be more true or plausible. Sociologically, Shuttleworth thinks we have a biased tendency to affirm, publish, or give attention to clever human-like animal stories, rather than focus on explaining the function and underlying processes of cognition, irrespective of humanity's relation. And I would have to agree. Framing the study of cognition and its evolution through an anthropomorphic lens has led some skeptics, like Clive Wine, to believe that we should not even pursue such questions in cognitive science with so obvious anthropomorphic properties. Why? Well, because there is a valid concern that the discourse of folk psychology, which is worryingly uncritical, will continue masquerading around as if it is a scientific explanation. If this is the case, then we cannot hope to offer an evolutionary plausible account of cognition. And if such skeptics are right, our current means of performing this task, like asking questions such as, do animals count as satisfying X, where X is some functional trait like a theory of mind, may be so considerably off track that we require radical change. I wish to deny this skeptical suggestion. I believe comparative cognitive ethology is not completely untenable, but a reframing of its aims, methods, and assumptions must contend with the issues raised. The pathway for doing so, according to Shuttleworth, requires that we move away from these naive anthropomorphic views of cognition to a project where comparative questions are best answered and approached by dissecting broad cognitive abilities into its elements or sub-processes. By deconstructing cognition this way, the sub-processes may demonstrate either wide continuity across species, discontinuity and uniqueness, or are confined to species influenced by a shared independent variable like similar ecology, social systems, or constraints. Shuttleworth recommends that the methodology should focus on a trait-by-trait analysis, and we must be open to the possibility that cognitive processes in human and animals alike will sometimes present as more human-like and sometimes more animal-like, but we cannot know this prior to gathering good evidence and subjecting it to rigorous phylogenetic testing. In part 2 of my thesis, I will examine a well-formulated phylogenetic inference practice that I think meets this challenge. A paper from McLean and colleagues I will lean on considerably describes a phylogenetic comparative approach. This methodology aims to integrate the efforts of comparative psychologists who probe the cognitive mechanisms and evolutionary biologists who have generated quantitative approaches for investigating the phylogenetic distribution and function of traits. Such a method, it is hoped, can provide guidance for testing and answering a variety of cognitive evolutionary questions such as those on the screen. Each of these questions has a corresponding method, but given our interest in human uniqueness, I will focus on question 1. To what degree phylogeny predicts the cognitive abilities of different taxa and its relation to question 2? To demonstrate the utility of these methods, McLean and colleagues recruited evidence from Amici et al. Studying inhibitory control, the ability to resist a prepotent behavior response in 12 primate species. Measuring involved recording whether such primates could resist instantly hitting a barrier when reaching for food. This trait is a nicely deconstructed target of explanation, avoiding the issues of anthropomorphism, yet appears to be an important precondition for solving more cognitively complex problems, suggestive of some divergence in cognitive evolution relevant to the human versus non-human animal debate. The results for this are on screen, with the scores of inhibitory control matched against a carefully constructed phylogenetic tree. In order to perform statistical methods on such phylogenetic data, accurate branch lengths need to reflect the lineage time divergence. Such trees are accessible through growing academic data sharing mechanisms and can even be constructed in cases where there is considerable uncertainty. The resemblance due to shared evolutionary history assumption, which pervades much of the comparative evolutionary research, rather than being merely asserted, can be measured for its degree of influence and truth. This occurs through a methodology called a phylogenetic signal. This process has been applied to various behavioral phenotypes, where phylogenetic inferences are safely made and is assumed to hold for comparative cognition. To identify the phylogenetic signal in the dataset, the application of phylogenetic statistical metrics proposed by Bloomberg occurs. Without exploring the details in much depth here, a lambda coefficient can estimate phylogenetic signal in the raw data. For the particular inhibitory controlled dataset, they found such a value to be approaching 0, which suggests more closely related species do not have more similar trait values in this particular case. Measuring the strength of association between shared history and shared signals of cognition can be informative. Lower phylogenetic signaling may suggest the possibility of any of the 4 conditions on screen. McLean believed their phylogenetic signal score was indicative of 0.4, that some independent variable or factor other than merely phylogeny influenced the differences in cognitive performance. This immediately enabled the testing of a new, more targeted hypothesis to occur. To investigate what this correlated factor may be, additional phylogenetic approaches can be used. However, given we have non-independence in the underlying data producing at least some phylogenetic signal, the correlation measure must be controlled with a special statistical measure called a PGLS test. The PGLS regression model constructed on the inhibitory control dataset was tested for whether it was correlated with relative brain size in the 12 primate species. The analysis revealed a trend towards species with relatively larger brains having greater inhibitory control, indicating a possible functional link between these traits. Importantly, the stronger the association between relative brain size and inhibitory control, or any paired comparison studies for that matter, like food storage and enhanced spatial memory, is dependent on the strength of the previously determined phylogenetic signal being funneled into the model. Thus, our confidence in making inferences about associations between cognitive traits and their evolution can be made more statistically significant if we incorporate phylogenetically informed research. This improved statistical power can detect real relationships and reduce the probability of erroneously inferring significance when no association exists. The major conclusion I want to draw is that a well-developed approach incorporating the findings of comparative psychology and statistical evolutionary methods is available. Following the phylogenetic comparative approach roughly means we must establish an accurate phylogenetic model, accrue enough data points representing species for comparison, provide a well-defined trait for analysis, and have good psychological experiments that operationalize and measure this trait across species. However, it is the application of rigorous statistical methods like the phylogenetic signal that, I argue, can ensure our phylogenetic inferences are plausibly identifying significant or non-significant relationships between phylogeny and the evolution of cognition. It's my belief that attending to these smaller scale processes can hopefully produce enough valid inferences that will enable us to decisively declare whether particular aspects of our cognition are continuous or discontinuous with the tree of life. Thank you for listening. I have some questions / worries on the screen, which I'd like you all to answer. Thank you.